Welcome to Stem Cell Technologies Educational Webinar. My name is Christy Fable and I'm a Product Manager here at Stem Cell Technologies. There will be two parts in today's conference, a 45-minute presentation segment and a 15-minute Q&A session. The Q&A period will be limited to 15 minutes. Any additional questions outside of this period may be addressed to Stem Cell Technologies via email or telephone. To ask a question during this presentation, please use the question box located to the right of the slides. In today's webinar, Dr. Brent Reynolds, Associate Professor at the McKnight Brain Institute and Director of the Adult Stem Cell Engineering and Therapeutic Core, will talk about the challenges associated with the identification and characterization of neural stem cells. Let me now hand you over to Dr. Reynolds.
Thank you, Christy, and thanks everyone for logging into the presentation, and a special thanks to Stem Cell Technologies for organizing and setting this up. So what I want to talk about today, based on the title, is identification, measurement, and application of neural stem cells. And why is this so difficult? I usually like to start my talks with this slide. This slide I think is very important for science in general. As scientists, we tend to be very attached to our hypotheses. We also tend to design our experiments to prove our hypotheses. And the statement by the famous physicist Richard Feynman is actually also more relevant when it comes to studying stem cells, because stem cells kind of have this elusive type quality and can be very difficult to study. So it's very important in designing your experiments, interpreting your experiments, that you actually don't fool yourself. And I think you'll see as I go through the talk of some times and places where we've actually fooled ourselves before, and hopefully we've corrected some of those, and hopefully we're not continuing to fool ourselves. Now, I would be remiss as a neurobiologist to not have a picture of Santiago Ramon y Cajal, the grandfather of modern-day neuroscience. And he had stated in the 1930s that once development has ended, everything may die, nothing may be regenerated. This was based on the data that he had at the time and became known as the no new neuron dogma. Now, this dogma was so strong that there was evidence in the 1950s, the 60s, the 70s, and the 1980s that new cell genesis was occurring in the adult mammalian brain and new neurogenesis was occurring. But those studies were largely ignored based on the strength of the dogma. But that dogma started to change in the early 1990s. And one of the things that contributed to the death of that dogma was a paper that Sam Weiss and I published in 1992, where we were able to isolate from the adult mammalian brain a cell that when we placed in tissue culture in a defined medium, so we didn't use any serum, and we used epidermal growth factor as the mitogen to drive its growth, caused the cell to divide and make a sphere or a little ball of cells that we called a neurosphere. Now, we could take one of those neurospheres, or we could take a population of those neurospheres, dissociate it into a single cell suspension, and it would generate more neurospheres. And we could continue to serially passage the cells like that, resulting in this arithmetic increase in the growth of the cells over time. Now, at any point in time, either in the early passages or in the later passages, we could take the cells, we could remove the epidermal growth factor, we could provide a substrate for the cells to sit down on, like laminin or polyornithine, and the cells would differentiate into the three primary cell types we find in the adult CNS, into the neurons, astrocytes, and oligodendrocytes. These cells had an absolute phenomenal proliferative potential, and in a series of experiments we had done now, getting close to 15 years ago now, is we had started with one adult neural stem cell, made one neurosphere. We passaged that neurosphere and got something like 50 to 100 new neurospheres, and then we continued to passage that population. Now, after several passages, we had more cells than we could actually, keep in our incubator, so we were then just passing an aliquot. But we passed the cells for 52 weeks, passaging them once a week. And had we been able to save all of the cells that we theoretically would have generated, we would have generated enough brain cells to repopulate every human brain on Earth a million times over. That's a phenomenal number of cells. That's up to an order of probably somewhere about 10 to the 50th number of cells. The cells did not become, or the proliferating population did not become growth factor independent. You remove the EGF and cells stop dividing. They retain their multipotency, and upon transplantation into the brains of host animals, they generate tumors. So this was a cell population, was totally under control, but a cell that could generate an absolutely phenomenal number of progeny. So we now know from some work that we've done, but work from a lot of other people in the community, where these cells are located. They line the ventricular system all the way from the very rostral portion to the very caudal portion. And we know from, and this is a slide from a collaborative project we had with the Ritz Lab when we were at the Queensland Brain Institute. And we also, where we basically mapped out from a rostral to caudal dimension the numbers of stem cells and progenitor cells. Now we also know from work from a lot of other labs that the neural stem cells, there's some hot spots in the brain, and the cell genesis that is occurring, those cells get incorporated into circuits and they become functional. And we know from work from other labs as well, this in particular this slide from Ole Lindvall's lab, that the cells, the stem cells and their progeny that are lying in the ventricles, when the brain is injured, they know the brain's been injured, they divide, and the progeny migrate to the site of injury in an attempt to repair the brain. So there are at least two hotspots of cell proliferation in the adult mammalian CNS, and that is in the subventricular zone, in the rostral portion of the brain, and in the hippocampus. We know that in these regions there are at least 9,000 cells being generated every day. We know that if you block cell genesis in the hippocampus, you have an effect on memory. We also know that if you place animals in a stimulating environment, that this increases neurogenesis. We know that exercise increases neurogenesis as well, and stress reduces neurogenesis. And drugs such as antidepressants have been demonstrated to increase neurogenesis. And even things like male pheromones can increase neurogenesis in the adult female brain. So there are stem cells in the adult rodent brain. These cells are generating new cells on a continual basis, and those progeny become functional cells, and we know that the environment and action can affect this process. But what about in the human brain? Is the same thing occurring? So a couple of years ago, there was a landmark paper from Maurice Curtis where they had described in the adult human brain a similar or an analogous region to the subventricular zone, and where cells were proliferating and migrating towards the olfactory bulb. And this slide that just came up is pCNA staining, and each one of those little dark dots is looking at a cell that is dividing. And when they quantitated that, at any point in time in that proliferative region, you have up to 100,000 cells that are dividing. So the reason this was such a landmark paper for me is that it demonstrates that all of the stuff that we're probably seeing and studying in the rodent brain is also occurring in the human brain, which means the manipulations that we do in the rodent brain may also have application in the human brain as well. So with this continual cell genesis in the rodent and human CNS resulting in the production of functional progeny and the observation that when the brain is injured, there's an attempt at repairing itself, together with our ability to isolate the cell and tissue culture that exhibits characteristics of a stem cell, together this all supports the notion that the adult mammalian brain has the ability to repair itself and that neural stem cells play a role in this process. So while it's been approximately 20 years since neural stem cells were isolated in tissue culture and it's been demonstrated this continual cell genesis is occurring, there has been a seemingly modest development made in two areas. One, in positively identifying neural stem cells and measuring their behavior. And two, in devising and implementing clinical strategies to encourage neural stem cell progeny to repair the CNS. So these are two questions that I want to focus on in the talk. The modest gains that have been seen is certainly not due to a lack of effort, as there's been a large community of outstanding scientists working on it, but rather the difficulty comes from the fact that stem cells in general are very difficult to study, and the brain is exceedingly complex to repair. So this can be broken down into two distinct problems, and I want to tell you about what we've been doing in the lab to address both of these problems. So I want to start with the issue of identifying and enumerating or measuring neural stem cells and their activity in tissue culture. And I want to start by actually giving you the answer now. And the answer to this is that a stem cell is a verb, not a noun. And I'll explain a little bit more about what I mean about that as I go on, but I think you'll get the point as I explain this situation. So unlike most other cell types, stem cells are identified not by what they look like, but by what they do. They're identified and characterized based on a functional definition. There are several reasons for this, with the two most relevant or obvious ones being that stem cells are rare, and number two, there is an absence of selective and exclusive markers to identify them. And so the key features of a stem cell, or neural stem cells in particular, are number one, cells proliferate, which is fairly obvious. Number two, they exhibit self-renewal over an extended period of time. And what I mean by this is that when a stem cell divides, it has a choice. It can divide asymmetrically, where it makes another stem cell and a non-stem cell, or maybe a progenitor cell. It can divide symmetrically, where it makes two stem cells, or the one thing it will not do, though, a stem cell won't divide and make two non-stem cells, because if it does that, it's actually not behaving or exhibiting this feature, or this key feature of a stem cell, which is self-renewal over an extended period of time. Now the next characteristic is the production of a large number of progeny. Now stem cells themselves don't divide a lot. As a matter of fact, they tend to be a fairly infrequently or slow cycling cell. And the reason it does this is to retain its proliferative potential over the lifespan of the animal, but rather it generates a large number of progeny through a transiently amplifying population, which takes on the bulk of the work of producing the large number of cells. Another feature is it has a multilineage differentiation potential. In other words, the tissue from which the stem cell is obtained, that cell will be responsible or have the capability of generating all of the cell types that you find in that tissue. And so in the brain, that's going to include neurons, astrocytes, and oligodendrocytes. So those are the main features of studying a stem cell in vitro. If you're studying stem cells in vivo, all of these characteristics apply. There is one additional feature, and that one additional feature is that the stem cell, you should be able to demonstrate that the stem cell can regenerate the tissue after injury. So, given that stem cells are defined by what they do, and we actually have a method to encourage stem cells to act in tissue culture, it would appear that we should be able to measure their frequency using the neurosphere assay. So we know the neurosphere assay can isolate and propagate neural stem cells, and that neural stem cells can form spheres. Hence, the number of stem cells should be enumerated from the numbers of spheres. So we've used this as a metric for a large number of years to enumerate neural stem cells, and in general, the community used it as well. But there's a problem with this, and the problem actually comes from this statement. Because a stem cell can form a neurosphere, therefore all neurospheres are derived from a stem cell. So in that statement, the first statement is actually true. A neural stem cell can form a neurosphere, but the second one is not. There was no evidence, at least when we started thinking about this a few years ago, that all neurospheres are derived from a stem cell. So we did the simple experiment of trying to passage individual neurospheres. And what we found was that it was only a small fraction, less than 10%, of the cells could be serially passaged to exhibit the key feature of extensive self-renewal and generation of a large number of progeny. So with that in mind, we started working on trying to develop assays that would allow us to better study and enumerate stem cell or neural stem cell frequency in tissue culture. And I want to tell you about these two assays. And the first assay is the neural colony forming cell assay that we did in collaboration with Sharon Lewis at Stem Cell Technologies. And the second assay is a mathematical algorithm that we've developed that allows us to look at symmetric stem cell divisions. So to start with, the first assay, the neural colony forming cell assay. So this uses a defined serum-free media mixed together with a semi-solid collagen-based media. And again, there's no serum involved and uses epidermal growth factor to drive the growth of the cells. When we plate cells at clonal density in this matrix or in this media together with the collagen, what we see, and we let the cells grow for anywhere from 21 to 28 days, is we see a whole bunch of different size colonies show up. And we see actually a really broad range, and you can get an idea where we see those sort of the grid lines there are 2 millimeters in diameter. So you can see we get some really large colonies, some that are, like up to 4 millimeters in diameter, and then we get a whole bunch of, we get small colonies as well. And one of the first questions we asked was, if we put the cells in this collagen matrix, does it affect the numbers of cells that have a proliferative potential? And the answer is no. We get the same sort of colony-forming frequency as sphere-forming frequency when we use the neurosphere assay. So there's no inhibition of cells that have a proliferative potential in these growth conditions. Second, we then enumerated the numbers of colonies that we saw. And what we saw is that the vast majority of colonies, approximately 90% of them, were less than 1 millimeter in diameter. And with the rest of the colonies being larger than that, and only a small fraction, approximately 3%, being quite large, and colonies that just appeared to continue to grow over time. All of the other colony sizes appeared to stop at some point. And so then we asked the question, out of all these colonies, how many of these can exhibit the key features of extensive self-renewal and generation of a large number of progeny? So we excised each one of those colonies or representatives from each grouping and compared and determined or looked at which ones could be serially passaged. And what we found was only the very large colonies that could continually be serially passaged that we could generate into a cell line. Also for the large colonies is they retained their multipotency as well. So after we had generated or serially passaged them, and we differentiated them, they continue to give rise to neurons, to astrocytes, and to oligodendrocytes. So when we look at the key in vitro stem cell characteristics, in the colony forming assay, the only colony size that was able to demonstrate the key features of proliferation, extensive self-renewal, generation of a large number of progeny, and a multilineage differentiation potential were the colonies that were over 2 millimeters in diameter. So what this meant then was that the numbers of spheres, now remember we would see approximately 3% of our cells would generate spheres, and then only 3% of those actually exhibited the characteristics of a stem cell. So the enumeration of spheres using the neurosphere assay overestimates neural stem cell frequency, as the majority of spheres are derived from a progenitor cell population. And the neural colony forming cell assay provides a simple one-step method to distinguish neural stem cells from progenitor cells based on their proliferative potential. And the final conclusion then of this would be that the neurosphere assay is an inappropriate method to enumerate neural stem cell frequency. However, there's a caveat to this, and I'd like to rechange that statement and state that the neurosphere assay as presently used is an inappropriate method to enumerate neural stem cell frequency. So there's something peculiar about the neurosphere assay. When you typically passage cells and cell lines, you break your cells up in some way, you plate them down. The vast majority, if not all the cells, survive and they start to proliferate. Your culture becomes confluent and you repeat that process. In the neurosphere assay, however, when you take your sphere and you break it into a single cell suspension and you plate them, most of the cells actually do not survive. Most of the cells actually die, And it's only the cells that divide that are going to make a sphere that survive. And so in our hands, that's about 3% of the population. So we can actually take advantage of this peculiarity in the neurosphere assay where the vast majority of the progeny die to look at the frequency of the numbers of symmetric cell divisions. And so what I want to do about the next 5 minutes is walk you through a thought experiment just to give you a bit of an idea of how we kind of came about this and kind of justifies this mathematical modeling approach. So in the first thought experiment, what we're going to do is we're going to keep the size of the sphere, the total numbers of cells, constant. There's going to be 2,000 cells in the sphere. But what we're going to change is going to be the numbers of stem cells in the sphere. So in this case, there's going to be one stem cell, and that stem cell never undergoes a symmetric division. So when we take that population of cells and we dissociate in a single cell suspension, 1,999 of the cells die. The one stem cell survives, it divides, and it makes another sphere of 2,000 cells, but with the containing only one stem cell. So when we plot that on a growth curve, what we see is we would get a flat growth curve. Now, if we change the thought experiment a little bit and say that we start with one stem cell, but when it makes a sphere, it undergoes one symmetric division so that we're going to have two stem cells within that sphere. Now, when I break up that sphere, and I'm going to get the vast majority of the cells die, except for the two stem cells, each one of those makes a sphere of equal numbers of cells, but each one of those has two stem cells in it. And then we're going just to continue to do this over time. And when we do this, what we see is we get a growth curve that looks something like this. Now, if we say, what if that stem cell that makes a sphere now undergoes two symmetric divisions? What happens? Well, then we see a growth curve that looks like this. And what if that stem cell undergoes three symmetric divisions? What type of growth curve would we get? Well, we see a growth curve that looks something like this. So what we can see from this example or this thought experiment is as the numbers of stem cells increases, remember that happens because the stem cell undergoes a symmetric division, that the slope of the growth curve is increasing. Now the first thing you think is, well, wait, that can't really be true. There must also be an influence of the progenitor cell population because that's the vast majority of the population. That must influence that growth curve. And yeah, it does influence the growth curve, but maybe not in the way that you may think. So in this thought experiment, what we're going to do is we're going to keep the numbers of stem cells in a sphere constant. It's always going to be 4. So it's going to be one stem cell. It will undergo 3 symmetric divisions, but we're going to change the numbers of cells in the sphere. And we're going to start with a stem cell that makes a sphere that contains 500 cells. Now, when I take that sphere and I break it up into a single cell suspension, I'm going to get 4 of my cells will survive, and each one of those cells will make a sphere containing 500 cells, and each sphere will have 4 stem cells in it. So if I plot my growth curve, what does my growth curve look like now? Well, it looks like that. Now I'm going to change. I'm going to say that each sphere, rather than having a total of 1,000 cells, is going to, or sorry, 500 cells is going to have 1,000 cells. And what does my growth curve look like? Well, it looks like this. Now, what if each sphere has 2,000 cells? What does my growth curve look like? Well, the growth curve looks like this. And so the slope of the growth curve doesn't change, but rather the elevation of the growth curve changes, and the y-intercept changes as well. So now we actually have a method that we can serially passage cells. We can have our control conditions. We can have some sort of treatment conditions. And we can take something as simple as looking at the slope of the line to estimate the frequency, not the frequency, but to estimate numbers of symmetric cell divisions. So we've been able then to take advantage of what is a peculiarity of the neurosphere assay, but what is also a defining feature of a stem cell. And that's the ability to exhibit extensive self-renewal or to be a long-term proliferating cell. Mix this together with some math, and we're able to focus in on this trait and extract some valuable information. So remember, stem cell has a choice. It can divide asymmetrically, or it can divide symmetrically. The more symmetric divisions the stem cell undergoes, the greater the increase in the slope of the curve. Now, people have been, so I guess, let's go back a sec. What we do then is we can then express this as a probability of a stem cell undergoing a symmetric division, or the frequency of numbers of symmetric divisions, and we can express this over any unit of time. We can say every 12 hours, 24 hours, 7 days, whatever we want. If you want to know more about this, Loic Dalroy, who was a postdoc in the lab, is now a faculty member at the University of Florida, had pioneered this work, and I would encourage you to read his paper and also to maybe contact him if there's any additional information. So, we've been studying, people study cell cycle kinetics. We've been doing this for a long time, usually using flow cytometry. But being able to, how do you use that sort of method to look at a relatively rare cell, at a cell that could be a 1 in 1,000, a 1 in 10,000. It's very difficult. But I think taking advantage of this peculiarity of the assay, of a bit of math as well, this has allowed us then to focus in on a feature of this cell and get some kind of handle on its behavior. So what I want to do is now give you some examples where we've used this in ourselves and in collaboration with other groups. So this is work that was done in Rod Ritz's lab when he was at the Queensland Brain Institute, and work that was principally done by Dan Blackmore. And Rod had noticed a few years ago that neurospheres derived from the adult CNS and fetal CNS have growth hormone receptor expression on them. So first question that was asked was, well, if we sort out the growth hormone receptor positive cells and the negative cells and compare them to control, do they exhibit stem cell characteristics? And the answer was yes. Second, if we looked at an animal that was a knockout for the growth hormone receptor, what sort of effect does that have on proliferation of this population? And what we found was that the slope of the growth curve was significantly reduced in the growth hormone receptor negative cells, suggesting that growth hormone receptor activation of that receptor has something to do with the stem cell's ability to undergo symmetric divisions. We could use the colony forming assay to look at those cells, serially passage cells in tissue culture. And on the right-hand side there, it compares the wild type to the growth hormone receptor knockout animals, looking at the numbers of larger cells that exhibit stem cell characteristics in tissue culture. And again, we see, as we would expect from the serially passage data, we see a reduction in the numbers of the stem cells. If we take wild-type animals and we add growth hormone into the cultures, we get an increase in the slope of the growth curve, suggesting those stem cells are undergoing more symmetric divisions. And if we look at that same population with colony-forming assay, we get data that supports the hypothesis that growth hormone is increasing, or activation of the receptor is increasing the number of neural stem cells by symmetric divisions. If we look directly and not culture the cells, but isolate cells directly from growth hormone receptor knockout animals and put it into the colony-forming assay, we get a significant reduction in the numbers of large colonies, suggesting these animals have less stem cells. And if we infuse growth hormone into control animals, and we then harvest the subventricular zone, and we look to see how many stem-like cells do we have based on tissue culture definition, we find that we get more large colonies, suggesting that in vivo growth hormone infusion causes more symmetric, or results in more symmetric divisions and an increase in the numbers of neural stem cells. And importantly, a relatively brief infusion of growth hormone, which gives us elevated numbers of neural stem cells based on our tissue culture definition of it, is actually retained over time, suggesting that growth hormone may play a role in increasing numbers of neural stem cells in vivo. Another place where we've looked at this has been looking at fetal, young adult, and older adult animals. And when we serially passage the cells, we see that there is a reduction in the slope of the growth curve over time. And if we look at the young adult animals and old adult animals, and we put the cells directly from the subventricular zone into the neural colony forming cell assay, we find that there is a reduction in the numbers of cells that can act like stem cells in tissue culture. Another place we've been using this has been in the studying brain tumors. And this is an MRI of a patient that has a glioblastoma. And we can take that tissue after they've surgically resected it, we can take a sample of it, and we can generate a cell line. So here's an example of 3 different cell lines, each of these derived from different patients with a glioblastoma, and we get different slopes of the growth curves. Now, we can express this as a probability or the frequency of the long-term proliferating cells, or AKA the cancer stem cells, being able to undergo symmetric divisions. And we see that there's significant differences between these 3 cell lines, between these 3 patients. And does this allow us to make any sort of predictions? And it does allow us to make a prediction of about how aggressive these cells are in vivo. So if we re-implant these, or if we implant these tumor cells, cell lines, into the or the frequency of undergoing symmetric stem cell divisions, we find that there's a very strong correlation. So the other place we've used this was looking at a molecule called TGF-beta. So TGF-beta is both a suppressor and a promoter of tumorigenesis. And it's expressed at high levels in some advanced cancers, including gliomas, where it's thought to drive the growth of the population and potentially of the stem cell population. So when we added TGF-beta to our neurosphere cultures and we counted the numbers of spheres that we would see, we'd find that TGF-beta caused a significant increase in the numbers of spheres. Now, prior to doing this work with the colony forming assay and the math model, we would have interpreted this as meaning that the TGF-beta was causing an increase in the numbers of stem cells. But when we serially passage cells that are continually exposed to the TGF-beta, we find that the frequency of the slope of the growth curve and the frequency and probability of undergoing a symmetric division is no different than it is in the control population. And then when we take cells that have been exposed to the TGF-beta for 4 passages in tissue culture and we implant them either sub-Q, the graph on the left side, or intracranial, the graph on the right side, there is no difference between those 2 populations. So what this leads us to believe, or to conclude then, is that while TGF-beta does play a role in driving tumor growth, the effect it has is not on the long-term proliferating, aka cancer stem cell population, but the effect is on the progenitor cell population. So why are stem cells so difficult to identify and measure? Well, 2 reasons. First, it's a rare cell type. It's really hard to study something that's very infrequent. And number 2 is there's an absence of markers that are specific and for the stem cell population and that are exclusive for this population as well. And so this is what led us to the statement that a stem cell is a verb. It's not a noun. So because stem cells are defined based on a functional criteria, we identify and study them based on their action. It's a little bit like that line in the movie Forrest Gump where Forrest's mother tells Forrest that stupid is as stupid does. Well, in this case, the statement would be that a stem cell is as a stem cell does. In other words, you can only identify a stem cell when it is acting like a stem cell. And if it's not doing so, even if in fact the cell you're studying is a stem cell, you're left having to conclude that the cell in question is not a stem cell. So I appreciate, well, this is all moderately confusing, and there's no really simple way out of this. I think this is one of the caveats of studying stem cells. Because of their elusive nature, because it's a functional definition, it is really difficult to do a definitive type experiment to identify that population. But I think it does help to explain then, why this population is so difficult to study in vivo and why we haven't seen huge progress in our ability to enumerate that cell population or definitively enumerate that cell population. So now I want to switch to a question. And while it's been like about 20 years since the method was used to identify neural stem cells in tissue culture to be able to expand this population and to grow literally endless quantities of these cells in tissue culture, there hasn't been a huge amount, or there are no clinical drugs or applications for neural stem cells in the clinic yet. And so it would seem that one of the obvious application would be using these cells for transplantation because we can generate large quantities of them. And while progress has been slow, there has been progress made. And so there are a number of entities that and labs that are working on this. So for instance, a company in California called Stem Cells Inc. has completed a couple phase 1 trials for pediatric disorders implanting neural stem cells. They're in a phase 1 trial for spinal cord injury in Switzerland. A company in Europe, in England, Reneuron, is in a phase 1/2 trial for stroke. And a company on the east coast of the U.S., Neuralstem, has started a phase 1 trial for ALS and will soon be entering spinal cord injury trial as well. There's also a number of academic investigators, for instance, Angelo Vescovi in Italy, who has been or who has just initiated a trial for implanting neural stem cells for ALS. But it's not so simple. It's not just an issue of having a whole bunch of these cells that you can grow up in tissue culture and implanting them and everything's going to work. And so here's a paper that came from a Swedish group where they were implanting neural stem cells and their progeny into spinal cord injured animals. And what they found was that the grafted stem cell progeny, while it did improve motor recovery, it also caused aberrant axonal sprouting, which led to hypersensitivity and pain-like symptoms. But they found that if they could prevent, by genetically expressing neurogenin 2, if they could prevent the astrocyte differentiation, they could prevent the graft-induced sprouting and hence the pain. So what this study raised the issue of is that transplanting into the injured spinal cord can have side effects and that there needs to be caution in moving forward in clinical trials. There's a second paper that came from a group in the US where they had implanted human neural stem cells into the adult spinal cord. And while they got fantastic differentiation, neuronal differentiation, one of the things that they noted at 3 to 6 months post-implantation was that about 3 to 5% of the cells were actually still dividing. And this is a little discerning in the sense that if you were putting these cells into a human patient that may live 20 or 30 years, if 3 to 5% of that population is going to divide for 20 or 30 years, that could turn out to be a potential problem. And then there was this paper, which it's a little hard to judge the results from this and actually what it means. But this was a child that had received several transplantations. It's unclear what they received transplants of, but they clearly did receive several transplantations of neural tissue, fetal neural tissue, which would have contained neural stem cells with it. They received literally millions, tens of millions of implanted cells, and the patient had developed there was some excessive proliferation and had a low-grade tumor. So in moving forward then, one of the things that we've been thinking about in the lab is this issue that if we're going to implant cells, we need to be able to isolate defined population of cells from renewable sources. And the reason this is important is, number one, it's going to be important for patient safety. We need to know what we're implanting. Number two, we need a reliable, consistent source of material, not just reliable that we have an undifferentiated population of neural stem cells in the progeny, but we actually need to know what percentage of different cell types we're implanting, and this is obviously going to be important for regulatory approval at some point in time. So neural stem cells, when you look at a sphere, is actually not made up of pure neural stem cells. It's actually made up of a mix of cells. This includes neural stem cells. It includes various types of progenitor cells. It includes cells that are partially differentiated into neurons, astrocytes, and oligodendrocytes. So where we wanted to start with is we said, well, we wanted to start with isolating a pure population. So we started with the question of, can we isolate a pure population of neurons? And so this is a project that Hassan Hazari, who worked on during his PhD and during his postdoc years with me, and he's now a faculty member back in Iran, had developed this method which we did in collaboration with the Steiner lab at the University of Florida, and we call the neuroblast assay. And it starts by plating cells and slightly growing up your neurospheres and plating cells onto a matrix. And what happens is we get astrocyte differentiation. You get this flat bed of astrocytes, and you see these progenitor cells that are layered on top. Now, over time, we watch those progenitor cells divide, and they start to form little clusters of cells. And over time, those clusters get larger and larger. And if we look what those clusters turn into, they turn into virtually neurons. If we take that population, we break it up into a single cell suspension, we put it on a flow cytometer, we see a plot that looks something like this. And within this particular experiment, we saw that approximately 10 to 15% of the cells were beta-tubulin positive. But 90% of those cells lived within one small area down in the bottom here. So we started to define, based on the FACS plot, two regions. One we call the P1 population, one a P2 population. And we knew that that P1 population appeared to be enriched for neurons. So if we use the sorter and we sort out the different populations and we look at the total population, we get a mixture of neurons and astrocytes. If we sort out just the P1 population, we get what appears to be an enriched beta-tubulin positive or neuronal population. And if we sort out the cells from the P2 population, we see what appears to be an enriched astrocyte population. We can quantitate this. And so here we can see that in the P1 population, approximately 75% of the cells are beta-tubulin positive. And if we look at the P2 population, it's around 5%, less than 10%. And when we look at the numbers of GFAP expressing cells, we get very few of them in the P population, but they're clearly enriched in that P2 population. So now we can look at the next question Hassan asked is there a way that I can increase the, that purity, which was at about 75%. So we found if you just isolated cells from the P1 population and he took out the O4 positive cells out of it, he could increase the purity to a little over 80%. If we took out the O4 and the A2B5 positive cells, he increased this to about 90%. But if he just pulled out the PSA NCAM positive cells out of the P1 population, he could get a 97% pure neuronal population. When you took these cells and you implanted them into an adult animal, and these are cells that came from a GFP expressing line in A. In B, you can, this is a stain for GFAB, and the reason you're getting reactive gliosis because this was the site or the implantation site. What we find is that the implanted cells, this purified neuronal population, do not revert into astrocytes. They retain their neuronal characteristics. And if we wait a little longer, like a month or two, and we look in these same animals and look at the cells that are implanted cells that are expressing GFAP, and we stain, the red stain here is for nu-N, for a mature neuron. What we see is we do see cells that are expressing GFAP. They're viable based on DAPI staining, and they're also expressing nu-N, suggesting that these cells will differentiate into mature neurons in the adult mammalian brain. The other thing we looked at was at survival factors. So we actually tried as hard as we could to force these cells to become astrocytes in tissue culture. And we did that with serum, and we did this with another molecule, BMP4. We could not force the cells to become astrocytes. But the interesting thing that came out of this is that we were able to identify BMP4 as being a very strong survival factor. And so this is what one of our implants would look like if we did not pre-treat the cells with anything. We get low survival. We don't get great process outgrowth. We don't appear to get great integration. But if we treat the cells with BMP4 for approximately 1 or 2 days before being implanted, what we find is we get greater process outgrowth of the cells and what appears to be better integration of them as well. We also find that we get an increase in the numbers of cells that survive and the numbers of surviving cells that express UN. And just another picture of cells that were pretreated with the BMP4, looking at their integration. And if we look out a month or 2 later and we do confocal microscopy, we see GFAP or GFP expressing cells, our donor cells expressing UN. So the other issue is this issue of abnormal proliferation. So if we're taking a neurosphere that has neural stem cells in it, now these neural stem cells in vivo are meant to live within particular niches. But if we're implanting those cells in parts of the brain where the neural stem cells are not meant to live, it is possible that one may get uncontrolled, not necessarily uncontrolled, but unwanted proliferation over time. And it's because the neurosphere, neural stem cell progeny, contain progenitors, stem cells, and differentiated cells as well. So first thing we looked at is when we did our sorting procedure and looked at our P1 and P2 and overall population, we did the neurosphere assay to say what's the proliferative potential of cells within each one of these populations. And we find that in the P1 population, the proliferative potential is significantly reduced. Then we used the colony forming assay to say what is the stem cell population within the P1 and P2 population. And it's very low. So in essence, we were able to, when we sort that neuronal population from the P1 population, we're able to take the true neural stem cell population from being 1 in 3,000 and take it to being 1 in 200,000. So while we're not able to get rid of all of the stem cell population, we do get rid of the vast majority of those cells that have that characteristic. So transplantation is rapidly moving towards the clinic. There's a number of requirements that we believe are going to be required for this to be successfully implemented. Number one, you clearly have to have a renewable source of cells. Number two, these cells have to be safe. There has to be no uncontrolled or no unwanted proliferation. And number three, you have to be able to control the dosing of these cells. And what I mean by that is that if you were doing, let's say, a phase 3 trial or a phase 2 or phase 3 trial for a drug, you need to know how much dose of the drug to actually give. You know how to do that because you know what the active compound is. So in the case of cell therapy, if you're implanting a mixed population of cells, your question becomes, which cell type or combination of cell types is actually having the effect? What's the mechanism? Is it the neurons, the astrocytes, is it the oligodendrocytes? Is it the mixture of all three, or maybe the mixture of 2 different populations? So you need to be able to control the dosing. To control the dosing, you need to be able to implant defined populations of cells. So we argue that neural stem cells can be a source of tissue for this. And number one, it's renewable. Number two, it can be safe because I think you can get rid of the vast majority of any cells that have any proliferative potential using some simple sorting methodologies. And number three, I think you can control the dosing because we can now isolate in a bin a pure population of neurons, we get a pure population of oligodendrocytes, or sorry, a relatively pure population of oligodendrocytes, we get a pure population of astrocytes. We can now start mixing those at various ratios and frequencies and implanting them to determine what is the right ratio to implant. Do we want to implant maybe 70% neurons, 20% astrocytes, and 10% oligodendrocytes? Or maybe you want 80% neurons and 20% astrocytes, or the other way around. We actually don't know what the effective cell type is, and we don't actually know what the right ratio is. But with this sorting methodology, I think we can start addressing those types of questions. So in summary, stem cells are located in the adult mammalian CNS. They're located in rodents, they're located in humans as well, and there appears to be an analogous region in the human CNS as there is in the rodent CNS. We can isolate cells in tissue culture using the neurosphere assay. This allows us to generate large quantities of cells, virtually unlimited quantities of cells that can be differentiated into the 3 cell types, into neurons, into astrocytes, and into oligodendrocytes. One of the problems, though, in studying neural stem cells is that stem cell is a verb. It's not a noun. So it made it very difficult, I think, to enumerate the numbers of cells because it's not as though it's kind of a thing. It's actually an action. And it's that action that we actually end up measuring. And so in regards to this, we've tried to develop a couple of assays which allow us to provide a better metric of measuring neural stem cells. And the first being the colony forming assay, and the second being the mathematical model, where we can look at the numbers of symmetric divisions that stem cells undergo in tissue culture. When it comes to application of neural stem cells, because we can grow large quantities of these in tissue culture, one of the applications then is for transplantation. We virtually have an unlimited numbers of cells in tissue culture that can be used as a donor source for implantation. But I think for this to move, for the field to move forward and to move forward quicker, is that we need to be able to isolate defined populations of differentiated cells, be it neurons, be it astrocytes, be it oligodendrocytes, and do the experiments where we determine what the correct ratio is for implanting. So I'd like to thank some of the people. There's been a lot of people involved in the data I presented to you today, but I really want to thank, from my lab, Maria Caldera, Will Dalroy, and Hassan Azari, who as a group were responsible for virtually all of the data that I showed you today. The Queensland Brain Institute, Rod Ritz, who we've collaborated on the math model, the colony forming assay, the growth hormone stuff, Kevin Burridge and Jeff Erickson, who were the mathematicians involved in the mathematical modeling study. And Stem Cell Technologies, Sharon Lewis, who helped develop the colony forming assay together with us. And at the University of Milan, Angelo Vescovi, who was a collaborator on many of these projects. So thank you, and I'll be happy to take any questions.
So thank you, Brent. That concludes the presentation segment of the webinar, and we will now begin our Q&A session by addressing the written questions that were submitted throughout the presentation. So first question, do you think we can use the same methods to isolate cancer stem cells in glioma?
So I think that we can use we have been using the mathematical model to look at the frequency of symmetric divisions of a long-term proliferating population within gliomas, or aka cancer stem cell population. And we are collaborating with people at Stem Cell Technologies with Sharon Lewis on trying to adapt the colony forming assay for working with tumor populations. If you take the colony forming assay as is and apply it to a glioma population, basically it doesn't work. Don't think it gives a valid readout, but we are in the middle of trying to develop it for that specific purpose.
Okay. So for the purpose of defining stem cell character, is in vivo tumor formation comparable to in vitro sphere formation?
Say that again.
So for the purpose of defining stem cell character, is in vivo tumor formation comparable to in vitro sphere formation?
Well, no, because those two things aren't necessarily the same. So for instance, we could take either human or we could take mouse neural stem cells. They can form spheres in tissue culture, but they don't form tumors in vivo at all. If we were to take a tumor population, well, what we have found, at least in our hands, is working on the GBM tumors, is every GBM tumor that we get a sample from surgery and that we can start a cell line, every one of those cell lines will form a tumor in an animal after being implanted. So I think there is some correlation between sphere formation of a tumor population and its ability to generate tumors in vivo, not for the somatic or normal stem cell population. I think Harvey Kornblum had a great paper a couple of years ago where he had published on a correlation between the outcomes or aggressiveness of glioblastoma and their ability to produce spheres in culture, and found a positive correlation.
Okay, so what do you think about the use of CD133 or prominence as a stem cell marker?
I think it's a fantastic marker. There's been a large number of papers that have looked at it as mostly as a marker for a cancer stem cell in gliomas, but also other types of tumors as well, so not just brain tumors. It's also been used as a marker for identifying neural stem cells. The only problem is that I don't think it is a selective or I don't think it's exclusive. So I don't think every cancer stem cell expresses CD133. And I think CD133 will also mark non-cancer stem cells as well. So it's not specific and it's not selective. But it doesn't mean it's not a great marker, because I think we've learned a lot about the biology of brain tumors based on use of that marker.
Is there any evidence for stem cells in the adult cerebellum?
During development, yes. During adult, I don't know. I don't believe there's been any from adult human, and there may be some evidence. You know what? I don't really have a good answer for that. I don't know if there is. I'm not really up on that literature. There may have been a paper that had demonstrated an adult stem cell in the cerebellum, but I don't think it's one of the hot spots like the subventricular zone, like the hippocampus, like the third ventricle, where we know there is continual cell genesis going on.
Okay. What do you define as the age of a young or an old adult brain?
So in that data I showed where we looked at young and old, our young animals were approximately 3 months old, and our old animals were between 18 and 20 months. So they were quite old, maybe between 18 and 24.
I'll go back to the top. Okay, sorry here. Has there been any studies using the neurosphere culture system to measure the frequency of neural stem cells or cancer stem cells in medulloblastoma cell populations?
Yes, there has been. There's been work done we've collaborated with Brandon Wainwright at the University of Queensland, and I'm blanking on his name, Dr. Rea, has also done some real pioneering work in this area. So the neurosphere assay can be used to isolate and expand the stem-like cells or cancer stem cells from those types of tumors.
Okay. Do you think that sphere size could be used as one of the criteria for characterizing cancer stem cells or normal spheres?
So for normal spheres, we actually thought that when we first realized that not every sphere was derived from a stem cell. And so we said, well, geez, maybe just the big spheres that do that. And in essence, there is some validity to that, because when we do the colony forming assay, what we're really doing is looking at the progeny of that single cell that made that colony, or a single cell that would make a sphere. The problem is you need to give it time to grow, to differentiate between, quote, the small colonies and the big colonies. So the colony forming assay, we let them grow for 3 to 4 weeks. If you do the sphere forming assay and you let the spheres grow for 3 to 4 weeks, two things happen. Number one, the spheres get so large that they start to become necrotic. And number two, the spheres start to come together in culture and start to fuse if you leave them that long. So it's been very difficult to use sphere size as a metric for the stem cell population. Rather, and this was kind of the whole point of doing the colony forming assay, the cells are in a fixed matrix. And it's interesting when you look at them under the microscope is what happens is they look like kind of little pockets of cells. So it's like you get a cell that will divide, migrate a little ways away, and then it will make a little colony of cells. And so those large colonies that I showed you, those ones that look over 2 millimeters in diameter, aren't like a mass that's tightly packed together, but it's rather loosely packed together, which then gives space for there to be exchange of oxygen and CO2, nutrients, and waste products as well. And I would argue it's going to be the same for the glioma cells. The problem if you're going to use glioma cells and use the neurosphere assay, and you want to measure the tumor-initiating cell based on size, you're going to run into the same problem. You're going to have to let them grow and get really big, and as they get really big, the whole dynamics change because I think you start to get a fair amount of cell death within the sphere, and you will get clumping and aggregation as well.
Okay, so there's a few more marker questions actually. So what do you think of using a combination of nestin and SOX2 to identify a neural stem cell?
So I think it's a great combination to use. The problem is that cells other than neural stem cells will express those as well. So this becomes this issue of a selective exclusive marker. Progenitor cells express nestin, and progenitor cells express SOX2 as well. So there's a great paper. This has kind of been what we call in the lab the Bible for solid tissue stem cells. It's a paper written in development in 1990, and it's by Ponton and Loeffler. And I would encourage everyone that you have to read this paper, you have to understand this paper. And they really go through the characteristics of solid tissue stem cells and how we define those solid tissue stem cells. And I think some of the points they make in that, when they made it, well over 20 years ago, are still pertinent today. They basically just got it right.
Okay, and so GFAP delta has been shown to mark neural stem cells in the subventricular zone. What is your opinion about this marker?
So I don't know a lot about that marker. We have collaborated with Ellie Hull's group in the past, and I think it's a great marker. The question is I don't know if it's, again, going to be an exclusive and specific marker. Does it mark the entire neural stem cell population? And does it mark only the neural stem cell population? I think it's a very relevant marker, and it's a lot of other markers that we have for looking at stem cells and neural stem cells. They tend not to mark the entire population, and they tend to mark other cells as well. But we're all doing the best we can, and it doesn't mean that just because a marker isn't perfect doesn't mean that the marker is not valuable. It gives us important information.
What do you think about the clonal or low-density assays for identifying stem cells?
So I think those clonality type assays are really important. It's one of the Evan Snyder had a really nice paper a few years ago in Nature Methods, where he had shown that if you grow cells at basically too high of a density, you will get cells will divide, they'll make spheres, but those spheres can aggregate. And that then ends up, if you're wanting to count numbers of spheres, we know not to count those anymore to look at stem cell population, but we do count numbers of spheres to look at what the proliferative population is, or the clonogenic frequency. If you do that at too low of a density, then your results are going to be mixed or probably not as valid because of that aggregation that happens. So doing things at a low or at a clonal density is absolutely important. One of the things we've noticed in doing some of those studies where we put one cell into a well is that there's a community effect with these cells. And as you start to isolate them, and once you start to reduce or get rid of that community, it changes the behavior of the cells as well. So, again, it's like everything we do in science. There is no perfect experiment. Everything has caveats. What's important is to be aware of those caveats. In other words, don't fool yourself when you're doing those experiments, and take what the limitations are into account when you're making the conclusions on the data you have.
Okay. Do you think adult neurogenesis is subject to age-associated regulation?
It appears to be. I believe the evidence from rodent studies is as animals age, cell genesis is reduced. It's probably not surprising. I think we see that in most of the tissues of the body where there's cell genesis. The functional capability of it doesn't necessarily just mean numbers of cells. The functional capability of cell genesis is affected as we age, and brain should be no different than any other tissue of the body.
Are you familiar with stem cells associated with the blood vessels in the brain?
Yeah, I've seen that work, and the work's excellent. I would think that that's probably true based on the data that we have. They may like that niche. Maybe they like it because it's a great source of nutrients just to be closer to, to be able to get rid of waste products to get fresh nutrients as well.
Okay, and what marker do you use in your lab when you want to characterize a neural stem cell?
Oh, hello?
Oh, sorry. What marker do you use in your lab when you want to characterize neural stem cells?
So we don't have any particular marker. Basically we use a functional criteria. So if you act like a stem cell, then we know that we actually had a stem cell population. We have used things like CD133. We've used, what's the other one I'm thinking of? CD15 is another one, looking at things like nestin expression, SOX2 expression. So we'll use as many of those markers as possible to support our hypothesis that we are looking at a stem cell population, but we don't have any one particular marker. Even the combination of those markers, again, it's not definitive, it's suggestive, it's helpful, it's the best we have to do that. I still think the best way to identify stem cells is based on how they're defined, which is a functional criteria. And I know that doesn't help in being able to look in your dish and say, or in your slice and say, that's a stem cell. But it's sort of, again, one of the limitations in studying the stem cells. They're very elusive, and we tend to define it on a functional definition, which means we need to look at it in a functional way.
So thank you, everyone. We are actually now out of time, but I would like to thank Brent Reynolds for a wonderful presentation and all of you for attending. We will have a recording of the webinar available on our website in a few weeks, and you can also visit stemcell.com for more information about our wide range of products to enrich, expand, and differentiate neural stem and progenitor cells. Thank you, everyone.
Okay, thank you, Chrissy.